Welcome, everybody. Are there any announcements? Are there any AgBio data announcements?
Probably the big one is the RCN was submitted.
That's right. Yeah, we submitted the RCN. Ava submitted it on Monday. I think it was Monday. Yeah, already a long week. So yeah, that's out. Thanks to everyone for
Huge
your
news.
comments, and we'll see how it's received.
Congratulations.
Yes, very good job. Excellent. Okay. That's that's great news. That's great news. So let me introduce Jack Gardiner. He's one of my coworkers. He works at Mays GDB. He's been there for at least the last 10 years. And Jack worked as a technician, and before he went back to graduate school, he got his PhD with EDCO, and then did a postdoc in Missouri. Is that right? No, Iowa State.
I did
And
it, not Iowa State, University of Iowa.
University of Iowa, and worked with Vicki Chandler for quite a long time, and now he's with us at Mace GDB, and he's going to talk about metabolomics.
Okay, thanks for the intro, Lisa. When Lisa asked me to talk about metabolomics, something I have some sort of fleeting experience with, I kind of had to decide how I really wanted to approach it, because there's just no way that I can really cover anything very thoroughly in 30 to 45 minutes that I wanted to talk. So this is going to be a very high level overview. And what I want you to come away with is that idea that maybe you understand a little bit about what metabolomics is, what it can do for you, and how it can help you to understand a living organism or an organismal system. There's a lot of facets to it, and it has tremendous potential, as yet to be fully tapped. So when I looked around for a definition of metabolomics, not surprisingly, there's a lot of different definitions floating around, but the one that I kind of liked was that it's a systematic study of unique chemical fingerprints that for specific cellular processes that specific cellular processes leave behind. And that seemed as good as any definition one could find in a sentence or two. And generally speaking, metabolomics is limited to molecules of 1.5 kilodaltons, and that should translate roughly to 1,500 grams per mole. So they're small molecules. There are some exceptions to this. Lipidomics, where they actually isolate protein lipid complexes, isolates much larger molecules. But for the most part, they're less than 1,500 grams or mole. Sometimes metabolomics is referred to as small molecule profiling, but I like the word metabolomics best. And it's always important to remember that a metabolic profile is just a mere snapshot in time. It's very fleeting. The metabolomic is extremely dynamic, and it changes from second to second. And in that regard, I looked at what some of the more cutting edge or future focused techniques were, and that seems to be focus on real-time metabolomics, which isn't necessarily applicable to a lot of people in this group, but it can measure hundreds of metabolites in a living cell in real time. This is mostly for single-cell organisms like yeast and bacteria, but I have no doubt that there's creative people in plant sciences that will be able to eventually figure out how to use this to ask questions that they're interested in. So what types of molecules are measured in metabolomics? And endogenous and exogenous are really terms that refer to more mammalian metabolomics, whereas in plants we tend to think of things in terms of primary and secondary metabolites. But endogenous are molecules produced within the organism, like what you would expect: amino acids, lipids, cofactors, nucleotides, modified sugars, hormones, that sort of thing. Whereas exogenous are produced external to the organism, and that's drugs, toxins, environmental contaminants in plants, maybe pesticides and herbicides. And no broad definition is without its controversy. So one of the controversies now is whether in the human microbiome, if you should include those bacteria as endogenous or exogenous, because they are free living, but they are living within the system and their whole life occurs within the system. So you can make arguments for they are either endogenous or exogenous. And this reflects my genetocentric view of metabolomics. Basically, I want to just mention early in the slide is that one of the, I think, real powers of metabolomics is that metabolites can be used as phenotypes. You know, if the experiment's properly designed, you definitely can map peaks to loci, you know, and of course this isn't trivial, but I really do believe that it can be done. And you can map lots of peaks to lots of loci. So it has the potential for being very powerful in a way that metabolomics is not powerful in and itself. The two of them, I guess that you would say gestalt, it's the sum is greater, the whole is greater than the sum of its parts. So by combining phenotyping and metabolomics. So in metabolomics, there's really two very broad approaches. There's the untargeted or shotgun profiling, if you will, and then there's the more classical targeted, which represents some analytical biochemistry, analytical biochemistry. So the untargeted really gives you the opportunity to see everything. You'll see a lot of some knowns, and even more unknowns, way more unknowns actually. And it tends to measure the relative abundance of compounds as opposed to the specific number of picograms per gram of dried tissue. As you might imagine, the analysis is just a lot more difficult. There's a lot more players. But as I mentioned in the previous slide, it's a very potentially powerful phenotyping tool. But in order for this to work, you need to have differences between the samples. And in the case of maize, this isn't so hard, but it may be harder in some organisms to get enough differences that it's worth the trouble to use it as a phenotyping tool. But whereas the targeted, you can optimize for specific metabolites like flavonoids, carotenoids, etc. You can quantify the actual amount of metabolites in a sample. In the data analysis, as you can imagine, less players is a lot easier. One downside is that standards are needed. So you do have to, and then the number of actual known compounds is really, in the big scheme of things, is really rather limited. So, but the targeted is amenable for things like looking at hormone metabolism or vitamin metabolism, or things like that, where there's 50 to 100 players involved. So when I started out this talk, I got to thinking about, and I'd always sort of wondered, but never really bothered to look into just how many metabolites are there in a sample. And so after doing some looking around and realizing just how complicated a question this really was, and how open-ended a question it really was, I came upon the Human Metabolomic Database, which, as the name implies, it's very focused on humans, and it's funded by Genome Canada, University of Alberta, I think, and the University of British Columbia. But I picked some samples, and they have a nice tool where you can actually search in it and look for the number of metabolites in samples. So I sort of put this together. Unfortunately, there weren't any good plant examples, but you can see the number of metabolites detected anywhere from 91 in sweat all the way up to 6,800 in feces. That's a lot of metabolites detected. This, I should say, this doesn't necessarily mean it's detected in one run or one spectra. This is just the number that they have detected. And then the column on the right represents just how many of them they're able to quantify. As you can see, it's much lower in human feces at 26% than, say, it is in cerebrospinal fluid at 909 or 99.5% of them are actually quantified. So there's really quite a range here, and human metabolomics database is really, as I learned, is really quite a research. I'll say a resource. I'll say a little bit more about that later in the talk. So I wanted to mention a word about statistical sampling and statistical and sampling considerations. It's a lot like microarrays. You want an experimental design, and that's very critical. At least 3 reps, and a lot of people say that 5 reps is better. Sample pooling is beneficial, but you have to be careful that you're carefully staging your samples. For some samples, that might be pretty easy, like a leaf in a specific part of the plant. But for other samples, like developing ears in maize, it's very difficult. And if you want to be careful when you pool samples that you don't pool out your variability. So it is beneficial, but it does require some attention to detail. And also, like microarrays, biological reps are preferred over technical reps. And so in terms of bang for your buck, it's better to look at more samples than the same sample many times. There's a lot of free software out there. It's dizzying how much free software is out there, but many people just go with a PCA, a percentile component analysis approach, especially in non-targeted metabolic profiling. At the cutting edge or leading edge is machine learning methods, and they've shown great promise in improving metabolite identification. It is important to remember, as one of the sources I was reading pointed out, metabolite, there is variation, substantial variation even in carefully collected samples. And this can really make principal component analysis challenging in some cases. And so the bottom line is that not all metabolites are all that tightly regulated. And this can be challenging to deal with this. I wanted to cover sort of in 4 slides, mind you. Each one of these slides could be a talk in and of itself. The metabolomics workflow, from sample extraction, collection, to separation, detection, and then data extraction. Sample extraction, an ideal sample extraction should be selective, cheap, simple, fast, reproducible, and then you want to introduce a metabolite quenching step to capture and stabilize the metabolites and in their native state. So this is actually a pretty important step in the sample prep. They're generally extracted with an organic solvent and then concentrated some way. If you're using a targeted approach, you probably can tweak it by just the choice of the organic solvent to separate what you're interested in away from what you're not interested in. Of course, then there's always the risk that when you do this, that you're throwing some baby away with the bathwater, and that some of these things you're throwing away might actually be interesting if you're trying to study. Generally, often in internal standards are added at the prep stage. So, for example, when I did GC chromatography on mammalian cancer cells, we added a plant steroid so that we could quantify our samples downstream. And it was nice having a plant steroid because we knew that wasn't present in mammalian cells. So we would add that at the very beginning of the extraction step. So then we really could back calculate just what the concentrations were of the various cancer cell lipids. And also, a last thing is that often there's a derivatization step, particularly when involved in gas chromatography. And this is treating your isolate with some sort of chemical that allows it to be volatilized as a gas, like in gas chromatography. This improves does improve the chromatography, and it also helps your column on your gas chromatography system, keeps it from degrading, but it does provide an additional step. And while I won't go into it here, there are issues with derivatization, and you have to be careful what you choose to as a derivatizer because it can confound some of the metabolites you see in your spectra. So once you've separated, or prepared your sample, it looks to me as that when I was looking at all these different methods, it really kind of falls down into most 90% of them probably fall down into three basic types of separation method. And they all sort of rely on the fact that you're separating what you've got into individual components. And this is prior to the next step, which is mass spec. You either use gas chromatography, which is really ideal for small and volatile molecules, and you can do derivatization to increase your profile. You can use HPLC, high performance liquid chromatography, or just liquid chromatography. This seems to be the most common method that people are using now. Although GC is still used a lot, it doesn't require, in general, any derivatization. You can look at a very wide range of molecules with great sensitivity. And then the last one is capillary electrophoresis. And most of us are familiar with electrophoresis from looking at DNA, RNA, and protein. So in this situation, in order to be amenable to capillary electrophoresis, it has to be a charged molecule. And I haven't seen a lot of examples of this, although I'm sure there are some out there. I just didn't catch them. Most of them seem to be either gas chromatography or HPLC method. So once you've separated your sample, the individual metabolites are usually detected by mass spectrometry, which essentially measures the mass to charge ratio, or the MZ ratio. I should say that sometimes NMR is used directly, but it seems to lack the sensitivity of mass spectrometry. Mass spectrometry could be the subject of many talks, and it's a very deep subject to try and discuss, but there's many types of mass spectrometry, and it seems that most of them end up measuring the mass to charge ratio in some way. There's electrospray, ionization, mass spectrometry, and there's MALDI, time of flight mass spectrometry. What's common to all mass spectrometers is that they have an ionization source to charge the molecule, a mass analyzer to measure it, and then, or to analyze the mass, and then a detector. And there's any of these three components have lots of variation, pages and pages of variation if you go into the literature. But all mass spectrometry, they are subject to either soft or hard ionization approach. In the soft approach, molecules are left intact. So if you inject a plant hormone in there, it still remains as a single molecule. In the hard, the ionization energy is high enough that the molecules are fragmented to produce a more detailed fingerprint. I think of this as opposed to, for people that might remember protein electrophoresis, there's one-dimensional, that would be soft protein electrophoresis, and then there's 2D protein electrophoresis. That would be equivalent to hard type of ionization. And I should say that a lot of folks actually couple this to tandem mass spectrometry, and essentially multiple rounds of mass spectrometry. And so this produces a really detailed fingerprint. It allows you to produce the kinds of to analyze hard ionization products. Typically what you do is you compare it to a standard. And it's very useful in the identification. I said here on the slide, unknown metabolites, but it's also useful in known metabolites, identifying them as well. It's just got a tremendous amount of information on each molecule that really allows you to, it's almost equivalent to what sequencing is to DNA. And there's the Metlin metabolite and chemical entity database at Scripps Research, and that contains 850,000 known standards that are used to assist in the identification of metabolites. I'm going to say a little bit more about that database later in the talk. So then once you've done your analysis and you've got everything resting on your hard drive, usually it comes down to the form of, if you're coupling metabolomics with mass spectrometry, or you have mass spectrometry, you extracted either as spectral values or metabolite features. And again, there's lots of free software for doing metabolites on the basis of either the retention time or the MZ value. But generally, it all boils down to is you end up having a treatment versus a compound table. So you might have your different treatments. Since I work in maize, I might think of your different mutants on one axis, and then the various compounds that are found in them on the other axis. You can use a variety of statistical or data reduction methods to ferret out statistically significant and hopefully meaningful biological differences. You don't really need to know what the compounds are, and for a lot of them, you never will know. But principal component, as I mentioned earlier, is often used. It's probably the most common method that's used in untargeted metabolomics. There is the use, some people use linear models, but these can be impacted by multicollinearity. And what I interpreted that to mean when I was preparing this talk is that there are certain metabolites that have basically can be fit to the same linear model, so it's hard to distinguish them. And then you can also apply your more traditional univariate and multivariate statistics, but you can use them, but you need to be careful to validate your models or correct for multiple comparisons and generate a false discovery rate. Machine learning approaches are increasingly being used in the last 5 years, and they're basically usually applied to retention time in the case of HPLC, and sometimes in the case of mass spec to the MZ values, and for prediction of the metabolites in a complex mixture. So when it gets into databases, there was a very nice research at the Metabolomics Society, and they had sort of broken it down into, I would say there's probably 50 databases devoted to metabolomics. And so it's sort of 3 broad categories. There's the comprehensive metabolomic databases, and they have a little bit of everything. In some cases, like in the human metabolomic database, they have a lot of everything. Then there's the more traditional pathway, metabolic pathway databases, and those are things like MetPsych, KEGG, HumanPsych, and BioPsych, and others. And then there's the compound-specific that are just databases, and they're devoted really to like one compound. Like there's the glycan database, or various other kind of compound databases. And then, as you might expect, there's the drug databases. They're devoted to both drugs and their breakdown products. And then there's the spectral databases, and these are really hardcore analytical chemistry databases, and they just are really probably more on the chemistry end of things than the biological end of things, and that's where people will deposit their spectra. And then my favorite is the last one, I saved for last, disease and physiology databases. And these are metabolic databases that are devoted to specific diseases or metabolomics on specific disease states. And so they don't there's not a lot of them, but they're very valuable. In this category, I would put the online inheritance in man database, which is devoted to the many human diseases. So I thought, since this was a pretty large topic, I would try and pick out 3 databases and describe them in a little bit of detail and pick them on the basis of just how different that they actually are. You can see here that I've chosen the Human Metabolome Database, Metabolites by EBI and Metlin at Scripps Research. The Human Metabolome Database contains information on about 114 metabolites that have been found in the human body that are actually identified. And so each of the 114 metabolites has a metabolo card, and that contains about 130 data fields, which about 2/3 of it's chemical data and clinical data, and about 1/3 to enzymatic and biochemistry data. And as I had mentioned, this is there's really a tremendous resource. It's funded by Genome Canada, and it's actually a part of suite additional database. There's Drug Bank, which has information on a little over 2,000 drugs and drug metabolites. There's the toxin database on 3,600 toxins and environmental pollutants. And then there's the pathway database on 25,000 metabolic and disease pathways. No plant stuff in here. And then there's the food database, which contains information on about 28,000 foods and more, and in my mind, more importantly, food additives. And it has both simple and sophisticated search. It has some molecular biology data, and so it has a sequence and blast search for that. It also has a text search of the text portion of the database, and an advanced search that allows structured queries of certain subfields. And then they have the spectral database search, where they have some spectra in the human metabolome database. Then the next one that probably someone like me working in the lab is most likely to come into contact with is metabolites, and it's really a database for where you want to maybe deposit your data if you're doing a small metabolomics study. It's an all-encompassing database in the sense that it focuses on the experiments and the data derived from them. It's both a repository and a resource, and it's highly curated metabolomics data. It's cross-species, some human metabolomics, as well as plant metabolomics experiments. Unfortunately, when I looked, there's only one maize experiments in there, but there's like 75 Arabidopsis experiments in there. So it is definitely a place, to put your data. It includes metabolite structures, reference spectra, cellular locations, roles, and experimental metadata. And it allows users to both, submit and browse. There's, as I think I mentioned, 778 studies. You can search by species, metabolite, or technique. You receive a stable identifier when you submit your data in a sort of a preformed format, and that's used for a publication reference. You can also share your data with your peer reviewers or collaborators. It's the preferred repository for a whole list of journals where they want you to put your data if you want to get published. And of course, not surprisingly, you can download complete studies as an archive. It also does have some training and education resources, both basic and advanced training. That's sort of associated with EBI. The last one database that I wanted to sort of draw attention to was the Metabolite and Chemical Entity Database. And it's the largest repository devoted to tandem mass spectrometry. It has about everything's done on the basis of standards. It has about 850,000 standards. And all the standards are characterized at multiple collision energies in both positive and negative ionization modes. And though I do not completely understand it, I think that the distinguishing between the negative and positive ionization is pretty critical. It produces an extremely detailed fingerprint. So if you want to identify a molecule, it's the place to go. It can be searched on the basis of spectra, mass, chemical formula, or structure. It's cloud-based, so that it's open and free to public, to researchers worldwide. And it was developed in the Sustack laboratory at Scripps Research. And it's really quite a resource if you're looking to identify compounds. But they estimate that, and when I looked at the latest paper, they estimate that even at 850,000 standards, that's only about 1% of the actual molecules out there. So There's lots of stuff. More is unknown than is known. So just wanted to make a couple of concluding remarks. Profiling is an evolving technology. A lot of it is at the ionization source and the detection methodology. It's still challenging. As I said, only 1% of all molecules are actually identified. Experimental design is and will remain critical. Sample pooling and replication are critical. Collect as much metadata as possible. You will never regret this. Yes, I know it's kind of tedious to collect it sometimes, but it will pay back. And before you even start your sample collection, consult a statistician to help you plan and design your experiment. You won't regret that either. Phenotyping combined with metabolic profiling is a very powerful combination, but you do need variation. My thinking is diversity panels, which identify GWAS, are preferred over biparental panels, simply from the fact that that you're just going to have more variation in a diversity panel. And untargeted approaches are preferred if possible, but they are challenging. And then, as we all know, databases will play a key role, are playing a key role now and will play one in the future. There's room for everybody. So that pretty much sums it all up. I could take questions and I could even try and answer them.
Thanks, Jack. Sounds so complicated.
It's very complicated, but it's powerful too.
Okay, Jack, I'm going to ask you the $64,000 question.
Oh no.
How are you going to link anything from metabolomics to anything else in Maze GDB?
Actually, some of it are already linked through Maze-Psych and Corn-Psych, no?
Yes, that's a tough question. I looked at taking in some metabolomics data at Maze GDB, and the honest answer is that we're not very set up very well to do this. I would like to tell you that we are. Maybe we could, like I think it was Tanner said, we could move to MetPsych or PlantPsych, link to that. I don't think we even have, to my knowledge, any real serious metabolomics data in Maze GDB at this time.
I could add, maybe this is Lisa, that Tanner, we do have in the corn psych and maize psych, the genes that are in the pathways are linked in Maze GDB. It's known, or if it's known, the pathway in which they participate is known. But we don't have pages for metabolites or anything like that. It's
Yeah.
really. I'm just wondering if we could co-op like the database in Canada that sounds really good. Could we just add plants and onto that or something, and then link back?
Well, I would say first we might be able to link to metabolites. Metabolites, the database, they might be open for us linking to specific studies. It's pretty clear that if you want to publish, that you're going to have to deposit your data, and it looks to me like the real place to publish your data for the kind of metabolomics we would be doing, targeted or untargeted, would be probably be metabolites.
Hi, this is Sushma. And so we work with the plant pathways in plant reactome and have developed also a couple of the plant psych type of databases for rice and monocots. So metabolites in psych and in both plant reactome, we use the PubChem and CABI
Mm-hmm.
as a source for metabolites. So we don't replicate metabolite pages. We link our metabolites to PubChem and CABI. And I hope that one of the places where this experimentally validated or identified metabolites can go is perhaps to PubChem or CABI. And the metabolites are actually already there. What somebody has to do is to add that line or the tag that this metabolite has been identified in such and such and such a species. You know?
Yes, and thanks for your comment there, because I should say that when I was looking at metabolites, they do have links to KEBI. And so that is probably exactly what we would probably want to do as the first step. There's no sense in duplicating the efforts of other people, and there's so much, there's more than enough to do. And yes, I think link to them.
If I may add,
like
Jack.
in UniProt, we have the gold standards for protein products, something that has been curated and something that has been predicted, and they use a gold star annotation for that. So the simple thing will be that CABI or PubChem can adopt to put a gold star if something is experimentally verified, and just add one column in their data that this metabolite has been identified in rice, soy, or whatever in humans. And when we bring that information in psych or plant reactome, then it automatically will be available, this information, to users if it is marked different color, maybe a gold standard or something.
Yes, and your thinking is that would be for known metabolites, but what if we just have a peak or something that we know is important, but we know maybe it's some kind of modified sterol, but we don't know any more than that.
So Jack, I think PubChem calls them substance. They have a substance database, which is obviously an NCBI funded effort internally from National Library of Medicine. So the PubChem stores all those information, like the mass and the spectral information that comes with that unknown substance from the shotgun assays, and the source, maybe more about tissue or plant or a species or things like that, and the experimental conditions or how the experiment was run. So that goes into the substance, so that people next time they can query that library if they match it, and then once it is identified what that actually is, then it goes to a next level of thing that you actually know something more about it. So that's how we are managing from the plant reactome perspective. PubChem is the one.
Yeah, that's good. That is something that I was not aware of. That I'm certainly no expert, but so thanks for your comment on that.
And so PubChem, what they do is they don't merge the records of, let's say there are two molecules or substances identified from two different sources, but they have the same spectra and things. They don't merge that data sets. They keep each data point from every individual source separately. So they have somewhat close to around 700 or 800 different sources, and they're actively curating millions of data points. And so, for example, if you collect the data from some source in maize, they will be able to add to the PubChem. That's how we shared our data, and the data is connected back and forth. And if there is a common reference with I believe what you saw shown was a metabolite or CABI, then obviously those are internal references in the PubChem already established through their other networking, other reference calls. So I hope, if I added to Sushma's thing, is that since we may or may not be the big experts in chemicals and small molecules aspects, we just borrow the detailed information from those sources, and they manage it. We just reference them using some stable identifier from our side.
Yeah, that sounds really useful.
Thanks.
I'm glad you mentioned.
Yeah, if you need it, we can put you in touch with the PubChem people.
Okay, and I'll take.
Yeah, I'd like to also add that we did a deep curation for the white psych for the researchers who work on grapes, and one of the grape researcher, he provided me list of 1,500 experimentally verified compounds, and we have actually added that in our psych database. And in psych, you can actually add metabolite, so there is a provision for that, and you can add also your node. So in some cases, when people are very interested in secondary metabolites in species like grapes or any other, one can curate easily and add that list. So we have done that as well. But always our reference is PubChem or KEBI, and we bring those molecules from there, but we add our note that it has been experimentally verified, and if it is published, then we link it to the publication.
This is Tanner. Perhaps as a follow-up, we can hear from Pankaj and Shushma in a different presentation about the plant metabolic databases. They have experience in both Reactome and Metapsych databases.
Yes, good.
We will be happy to share that or make a presentation.
Well, I'd be certainly interested. My background is more in the biochemistry and the analytical stuff, and probably not so much in the how the information is stored. And so that would be interesting a presentation for me.
Well, we certainly have slots, so we can do that in a couple months. Yeah.
Sure, and we'll be happy to invite, if we can, to ask the PubChem guys to give us a joint presentation or something like that, because it's a common topic, right?
Yeah.
Yeah, so we can do half and half, what we have in plant reactome and our site databases, and then Pub can provide their own perspective on that.
And are there any else of you that do you connect metabolites to gene expression or any in your databases that are all represented here?
Like in GDR and our other databases, the metabolite data that we have is through QTL papers, especially in Rosaceae, because we deal with fruits. A lot of QTL studies deal with like measuring metabolites. So how we deal with is just making those metabolite names as a Rosaceae trait ontology, because it's for QTLs we do it that way. So I would be interested in how quickly we can connect those trait names. I mean, metabolite name is basically a trait name in our database for those type of study. Like it would be nice to link to like metabolite database or something like that. I wonder, KEBI, I know trait ontology people have been trying to link like appropriate trait ontology terms to KEBI, so we can do something like that. But then we can link to other databases, like, but I haven't heard of metabolite database before, so. Yeah, I don't know if there's an easy way to like download or search, like to link up to our names or something, to make hyperlinks.
So that's a good idea, because I think you have in the ontological format, and Plantium does have the mapping
to KEBI
aspects. Right, yeah. So some of our trait terms, as you mentioned, that are composites of biochemical small molecule entity and quality. Mm-hmm. So in that case, the biochemical entity is actually referred or pulled or mapped to the KEBI. And if we don't have that entity in KEBI, for example, because if they're encountering it for the first time, they're very, very responsive. in that sense. But I'll give you what I understood from the last time my conversation is Kebi is one aspect, but I think Campbell is the one which is taking over because Kebi is, I think it's going to be merged or something. Campbell is taking over.
How do you spell
That's much bigger. CHEMBL.
Oh, okay.
Yeah. But
So
obviously those things are maintained.
Yeah, I like to add that in plant reactome, we have a provision if you have a metabolite data and you have the metabolite IDs from PubChem or Kebi, you can actually upload your data and you can do the expression analysis of metabolites in different samples in context of the plant pathways.
Okay.
So that is something new that plant reactome offers. So you can upload like the gene expression data. I'm not sure if they have updated and made it available for doing the metabolite, but we do have that in plant reactome.
Okay.
More questions? More comments? More thoughts?
So you're saying that you can do an omics, what in the psych databases, an omics overlay onto the pathways in plant reactome?
Yes, in plant reactome you can do both. You can upload the transcriptome data, but you can also upload proteome or metabolome data. The only thing you have to do in your Excel file, you have to make sure that instead of gene ID, you use the metabolite ID. And so each metabolite has an individual ID in PubChem and Kebi, so you can choose either of those. So as long as you have your metabolite, you have your metabolite ID, and you have the expression profile in different samples, you can do the omic analysis.
Rex, also something to think about with MetaPsych databases, you can sometimes do more when you download it as a local copy than what you present through web. So you can do this overlays easier on your local copy for MetaPsych.
Visualization options on the desktop copy are slightly different. You can generate slightly better graphics when you have the desktop copy. And then also the other feature is Smart Table also. I think it is a very good feature that they have added more recently.
So Jack, that was a very good presentation, and I would add, means I think you were presenting that there are not much of crop plant or other plant data other than Arabidopsis that's available in the public domain, and my observation is that quite a lot of this data is also proprietary. A lot of labs maintain their internal repository and databases, but not necessarily public. So maybe one question for our group is how do we motivate and what could be the guidelines or something like that to think about how do we want to navigate this area for the plant community to open up their data, because spectral data is more proprietary and not many people are willing to open that data. There's a lot of it in several good labs. I won't name them, but there's a lot of off the chart data sets.
Yeah, we had someone talk to us a few months ago, to the MACE GB team. And the sense I got from him was that he was not satisfied with any of the existing databases. But I don't remember his reasons, why, none of the existing ones met his needs. And it may have been partly because he thought, well, you can put your data there and you can get it all out, but you can't do much more than that with it.
So the problem is that people want to use databases, but they don't want to share their data with others. So I work with the great folks, and a lot of them, they work with company, and nobody wants that data to be publicly made available.
Yeah, I think it's getting better. It's still a problem, but I think it's getting better. But it could also be, just thinking aloud here, the metabolite data is not cheap to produce. And it seems like it can be mined for a lot. And some people could be reluctant to put it out there in public until they feel like they've completely mined it.
I'm sure that, Althea, that's the case in industry. They're not so much worried about you and me. They're worried about their competitors. Like Pioneer might be worried about Monsanto, or just certain kinds of data would indicate what they're working on. And they probably don't really want people to know that.
Yeah, well, I'm not surprised that the private companies wouldn't share, but there may be reasons why labs would be reluctant, aside from, not realizing it matters.
Well, my experience from microarrays is that you have to make it painfully easy for them to submit their data.
And that too, yes.
Otherwise, and you know this from Plex probably, otherwise it ain't going to happen.
Yeah, and as you pointed out, there is a lot of metadata.
Yeah.
So do you think that maybe bringing in Lloyd Sumner from University of Missouri, I think, to give us some spiel about what the community is thinking about, because he's one of the pioneers and founders of that organization, or which deals with the metabolite community, and chances are that they may have some insights. Oliver Feen at UC Davis and Lloyd Sumner.
Yeah, that would be great. We're all sort of stuck in the genomic world, and I'm worried we're going to be stuck.
That's true.
So I think it's going to look at this other data.
Last I spoke with them is that there is no funded plant repository or ag bio repository for that kind of data. But if there are other publicly available, that's absolutely fine. But do they meet the needs of the community to open up their data? So maybe having a good conversation with that organization would be a better idea.
How about, so we're sort of plant-centric here, but there are some animals in our group. So Jack, did you see many, or did you check for, livestock samples?
I didn't. I did see the group at Davis. Looks like they have a very good metabolomics group there. But I didn't see a lot of livestock metabolomics. I didn't tend to dive into particular, because I was so broad, I probably could have missed it. But no, I didn't. And when I looked for a kind of a central plant repository, metabolites was really all I could come up with.
Yeah, and also I'm thinking that livestock are a lot more like people than plants are. So what works for humans may work for livestock, but maybe not for plants.
Mm-hmm.
Yeah, and what I'm realizing is also sometimes these metabolite profiles actually very quickly turn into the proprietary aspects, because people are trying to target it using drugs or some engineering aspects, or pathway engineering. So that's one of the main reasons. For example, in cow, buffalo, and other breast milk, you will see some certain glycosides. Those are very beneficial for health and microbiome aspects. And we absolutely have no data available in public on those kinds of stuff. So yeah, it's a different world, but I think bringing in the conversation and bringing those community members on board would actually help, including microbiomes.
Well, great. We're coming up to our hour here. Maybe when we talk next time, what I'm seeing in the literature on metabolomics is people are doing their RNA-seq and this and that and other omics, and then they do some metabolomics. So right now, we're not capturing that data because it's we don't know exactly how to do it, but maybe we could take curators. We could have a little group of curators and figure out if any of that data should be captured.
I think a good place to start might be, like was suggested, is trying to bring in some other speakers that have a deeper knowledge of the subject than I or others of us, and just as to what are they doing,
Yeah, well,
The group at UC Davis looked promising because I think they were sort of they were at least focused some on plants, if I remember right.
Yeah, Oliver Feen was on rice and rhabdopsin, I guess.
yeah.
Maybe corn. And Lloyd Sumner at Missouri, I think he's corn as well, corn and soy.
Oh, okay.
Well, Pankaj and Shushma, I'll get in touch with you about a talk later this year. Next month, we're going to have Peithin Zhang. She's from Phoenix Bioinformatics and is working on phylogenes, which is a phylogenetic tree of genes, and she's doing literature curation to get more precise Go annotations on members of gene families. So I'm looking forward to hearing her talk. And anything else before we go? Congratulations on getting the RCN grant in. That's a milestone. Thank you very much, Jack. That was very informative, and really appreciate you taking the time to do that. And I'll see you next month.